Yeah, so first off, thanks for coming back from the open bar. I'm sure that was tough, guys. My name is Dr. Michael Johnson. I'm the CEO and co-founder of Visikol, and the focus of my talk today is all around turning tissues into data. So since the inception of the microscope over a century ago, the way that we've characterized tissues, whether it's a biopsy from a clinical cancer case, whether it's a mouse brain, any type of tissue, the way that we've characterized it is we take a tissue out of a patient, out of an animal, we take a three-dimensional tissue, we slice it into a few ultra-thin two-dimensional slices, we put it on a glass microscope slide like you remember from high school biology class, we stain it, and then a pathologist or researcher looks at that slide on a microscope and by eye makes an evaluation of that slide. So they're diagnosing disease, which ultimately results in a treatment for a patient, or they're determining what's going on within a tissue for a drug discovery effort, or just a basic research question. This is the paradigm for imaging tissues as it stands today. Now, what I'm going to talk about are the two major limitations with this approach and what our company is actually focused on addressing. So the first of which is that less than 1% of the data from a tissue is actually used today. So we take out a biopsy from a patient, which is part of a tumor. We're only taking a few ultra-thin two-dimensional slices from that tissue. Now, this is very problematic because if I have a biopsy, which is really complex, it's really heterogeneous tissue, I could have a biomarker here, another one here that's very important, and if I take a two-dimensional slice over here, I'm missing that information. So today we bias data by taking just a little bit of the information that's embodied within these tissues. The other limitation is that today a human is looking at these slides, hundreds of slides per day, slide after slide all day long, and there's a lot of variability in how they look at the slide as the day goes on. There's variability amongst different pathologists, and the main limitation here is that we're only looking for a few features within each one of these slides, maybe one or two or three, but there's thousands and thousands of features that are in each one of these images that today go missed. Now, the problem is that this paradigm for many clinical applications leads to diagnosis. In some cases, we have as high as 25% of cases being misdiagnosed. This is for really heterogeneous and complex types of breast cancer, but this results in patients having poor outcomes, the wrong treatment options coming out of the diagnosis, and ultimately patients aren't getting the type of treatment they need. In the research space, this results in drugs going to market which may or may not be good drugs, which have high toxicity that's missed by a pathologist, or features that are just never seen. Now, the future to the people in this room is quite obvious, but this is a slow field. This has been done this way for a century, and disruption in this field hasn't taken place in the way it has in a lot of other fields with image analysis. But what our company is focused on is imaging tissues in 3D. So getting rid of a few slides and pulling a little bit of data from tissues and actually getting all the data from a tissue. So behind me, we have a lung biopsy from a person that we pulled out. And what we're doing is we're imaging through that entire lung biopsy and actually capturing all of the information present within that tissue, all of that data, instead of just a tiny fraction of the data embodied within that tissue. The other place that we're going in this space is quantitative image analysis. This is new in this space, just pulling out a few features here and there, looking at nuclei, looking at different cell types. That is new in this field. But there's a huge shift coming in this field from going from the qualitative approach of having a person look at slide after slide and moving towards taking out hundreds and thousands of features from these tissues and mining those features for actionable insights. And what truly excites me about this field and where this field is going is data mining. Today, we pull out a few features. When we look at a biopsy from a patient, that report turns into a few sentences on a piece of paper that doesn't go anywhere. With taking an approach of getting all the data and quantitatively looking at that data, we can actually pull out thousands and thousands of features from tissues, and we can use that with clinical outcome data to actually prescribe the best treatment for a patient's specific type of cancer. And the future where I see this going is deciding for a biopsy based upon the features within that biopsy that we're extracting, saying that this drug and that drug work best for you. So there's a lot of advantages of image analysis in this space, but there's major disruptions coming in the next 5 to 10 years in this field. Thank you.
Fantastic. Thank you very much. Well done, Michael.